in a way, my presentation will go on again speaking about mitochondria and calcium and taking up a question which is going around since many, many years. I would say since 60 years ago, when it was discovered that mitochondria can take up calcium. The question that how much is important this calcium taken up from the mitochondria for the cytosol, not for the mitochondria itself, but for the cytosol around. And in many cell types, there is clear indication that mitochondria can affect cytosolic calcium. In skeletal muscle, the point is rather questionable, rather doubtful, also because mitochondria are not so many as in other cell types, for example, in cardiomyocyte, and calcium released by the sarcoplasmic reticulum is very much greater than in other cell types. But just to refer to three, not recent, but anyway, not so old papers, just very briefly. Gillet, 97, so more than 20 years ago, showed that blocking the entrance of calcium in the mitochondria with ruthenium red, it could prolong the contraction, delay the relaxation. That is, calcium remain longer in the sarcoplasmic reticulum. More or less the same observation was confirmed in 2010 by Weiss et al., the group of Bruno Jacquemond in Lyon. They showed that depolarizing the mitochondria with FCCP, they could prolong a calcium transient. So keep the calcium longer in the cytosol. And in the same years, in the lab of Rios, E et al. Published a paper using a special type of transgenic mice, G93 mice, which is a model used also for ALS, where mitochondria are depolarized only in some part of the fiber. So there is a part of the fiber which is depolarized and the mitochondria don't work, and a part of the fiber where mitochondria are normally polarized and they are active and as normal. Well, during an electrical stimulation voltage lamp of 500 milliseconds, the calcium transient was with a greater amplitude and longer duration where the mitochondria were not working. And again, suggesting that mitochondria can regulate the cytosolic calcium level. Cytosolic calcium level is variable in the muscle fibers because of the interaction between sarcoplasmic reticulum, which releases calcium, and several mechanisms to remove calcium from the cytosol. So in this cartoon, we can see on the left side terminal cistern, electrical stimulation or chemical stimulation, for example, caffeine, can produce opening of the iodine receptor, calcium enters in the cytosol, the concentration goes up quickly, but immediately goes down. So it's a transient variation. Why it goes down? Because calcium is taken up by troponin C, producing then the contraction, by parvalbumin, calmodulin, and then by transport mechanism like MCU, which brings calcium in the mitochondrial matrix, SERCA, which brings calcium back to the sarcoplasmic reticulum, and sodium calcium exchanger and sarcolemmal pump, which brings calcium outside to extracellular medium, intercellular environment. Well, why underlined in red two of these factors, parvalbumin and mitochondria? Because what we are going to consider now is just the interaction between these two factors playing on the cytosolic calcium, parvalbumin and mitochondria. And we studied the interaction between these two factors of lowering calcium, acting on this part, not on the release, but on the removal, using comparison between three different conditions. In three cases, we studied single fiber dissociated from FDB muscle of the mice, and we compared wild-type fiber from C56 black 6 mice, fibers from same strain of mice where there was a constitutive ablation of the parvalbumin gene, so no parvalbumin present, and fibers where a transient downregulation of parvalbumin, maybe not completely ablation, but anyway, at least downregulation was obtained by silencing RNA with transfection of a plasmid carrying with a short pin RNA and also a fluorescent marker to identify the fibers efficiently transfected with the plasmid. So a comparison between these three conditions. Well, if we must say what was our expectation, removing completely or partially parvalbumin, is that if the more abundant buffer of the calcium in the cytosol is removed, the concentration of calcium will be higher in the cytosol. We have simulated this with a model, and a model which can predict rather well the movement of calcium between three compartments, the cytosol, the sarcoplasmic reticulum, and the mitochondria. Simulate, reproduce rather well what happens in this case, as you can see here in the middle, with tetanic stimulation at 60 Hz, so not completely fused, lasting for 2 seconds. We can simulate rather well this. If we simulate the condition where parvalbumin is removed and nothing else is changed, what we obtained is that the concentration in the cytosol is more or less doubled from this point from this level to this level. This is the expectation. We remove a buffer, the concentration will be higher. But the observation is not in this direction. It's not in this direction because what we can observe is that the amplitude of the calcium transient is not significantly changed. Actually, it's not significantly changed the resting level. Here we see the resting condition, the resting level concentration of calcium measured with 4:2 ratio. Then when we stimulate for 2 seconds, 60 Hz, produce an unfused tetany, we have an increase which is as an amplitude rather similar in wild type and knockout. No parvalbumin, no increase. There is some minor change in the relaxation phase, but the amplitude is not changed. So what can be the explanation of this? It means that there is something else which contributes to remove calcium if parvalbumin cannot do anymore its job. We confirm this result also challenging the cytosolic calcium with transient induced by caffeine. 10 millimolar caffeine produce almost a maximal, not really maximal, but very big release of calcium from the sarcoplasmic reticulum. Again, the amplitude of its transient, black and gray, was very similar in wild type, and after removal of calsequestrin, in this parvalbumin, in this case, we compared also the constitutive ablation, the knockout, and the transient downregulation with silencing of the parvalbumin gene expression. So there is something which can compensate for the lack of parvalbumin. The first possibility we considered was that the sarcoplasmic reticulum was releasing less calcium. For this reason, we went to measure the calcium concentration, the free calcium concentration inside the lumen of the sarcoplasmic reticulum using a chameleon targeted to the sarcoplasmic reticulum, ER-D1 chameleon. Well, the resting basal level was not changed. And even during our tetanic stimulation, we take this as a model to produce a significant release of calcium, we could observe a decrease of calcium concentration, some minor depletion, but no difference between wild type and knockout mice. Since the majority of calcium in the sarcoplasmic reticulum is not free, but is bound to buffer proteins in the first place, calsequestrin, with a ratio 34 times calcium bound to calcium free, we also checked the expression of calsequestrin as RNA and as proteins. There was no difference. So apparently, sarcoplasmic reticulum is still working as in wild-type mice. Then we moved to the mitochondria. And we measure calcium inside the mitochondria. In this case, we use as a fluorescent probe the GCAMP targeted to the mitochondrial matrix. And in the mitochondria, in the matrix of the mitochondria, we observe that in resting condition, the concentration of calcium was similar in wild type and in knockout, and also in silence and fibers. But during stimulation, either with caffeine or electrical stimulation, caffeine on the left and electrical stimulation on the right side, there was an increase in calcium concentration about 2 times what was observed in well-type fibers. So it means that the mitochondria can take up a considerable amount of calcium. And this is just the free calcium that we see. And in the mitochondria, the ratio between the free and the bound calcium is estimated to be in the order between 100 and 200 times. So a significant amount of calcium is taken up by the mitochondria. With the help of Feliciano and Simone, we observed also the volume of the mitochondrial compartment. The volume of the mitochondrial compartment was increased by about 30% due to the combination of the increase of the mitochondrial number and the increase of the mitochondrial size. In addition, the number of the mitochondria close to the calcium release unit was increased when parvalbumin was removed. And this is also an important aspect. So the contribution of the mitochondria to control cytosolic calcium seems to be due to the combination of more calcium taken up by the mitochondria and more mitochondria volume available to accumulate calcium. So this seems a good candidate for the explanation that parvalbumin removal is not so dramatic effect as one can expect. Mitochondria can replace, at least in part, the role of the parvalbumin. But to check with an experimental approach this, we did another experiment. We transfected in the same muscle, at the same time, plasmid to silence parvalbumin gene and silence to the MCU gene. MCU is responsible for the biggest part of the calcium entering the mitochondria. So when MCU is silenced, the calcium taken up by the mitochondria is significantly decreased. The efficiency of the transfection, plasmidic transfection, is never 100%. So in the same muscle, in the same FDB muscle, we could find 4 different fiber populations. Fiber populations which were not transfected neither by MCU silencing plasmid, neither by the parvalbumin silencing plasmid. Therefore, they were black, no fluorescence. Then fibers which were transfected by the plasmid to silence MCU, carrying also a cherry fluorescent protein to make them visible, that's red color. Fibers silenced, transfected with the plasmid to silence parvalbumin, carrying also a GFP protein to get identification with the green color. And then there was a fiber which were transfected by both plasmid, and therefore they appeared in yellow. As you can see here in the lower panel, you can see some part completely black, some part yellow, some part green, and also some few fibers in red. So we have in the same muscle availability of 4 different types of fibers. Well, in this fiber, we could measure the mitochondrial calcium and the cytosolic calcium. The mitochondrial calcium taken up by fibers where MCU was silenced, either wild type or with parvalbumin silencing, showed a reduction of the level of calcium in the matrix. So silencing the MCU, less calcium entering the mitochondria in all cases. But when we look at the cytosolic calcium, what we could observe, that compared to wild type, the black, the silencing on only MCU doesn't affect increase the amplitude of the transient induced by caffeine 10 millimolar, so big release of calcium from the sarcoplasmic reticulum. Silencing only of parvalbumin, the dashed line, also doesn't affect, but silencing both parvalbumin and MCU cause a big increase in the amplitude. It means that when we block the calcium taken up by the mitochondria, cytosolic calcium is out of control, is not anymore controlled. So mitochondria actually can contribute, in this case, to regulate, to control the cytosolic calcium level. So we can conclude, and we have summarized in 4 points. Parvalbumin removal from the cytosol does not alter free calcium at rest and after release induced by electrical stimulation or caffeine administration. So there is something else playing a role. Parvalbumin removal is accompanied by a significantly greater calcium taken up, accumulated in the mitochondria. If we silence MCU, we reduce the mitochondrial calcium uptake, and when we combine the silencing of MCU with silencing of parvalbumin, there will be an increase of calcium in the cytosol. Thus, we can conclude that in the particular case when parvalbumin doesn't play its role of buffering the calcium, mitochondria can contribute significantly to control the cytosolic calcium level. And that's all the story. Thank you very much for attention.
Thank you, Carlo, for this beautiful presentation. I knew already a lot of this data, obviously.
Of course.
And the session is open for discussion. There is some questions from the audience.
No, I'm involved in the work, so ask
I know.
You're involved in the work.
I want to ask Carlo. Carlo,
yeah.
Give me a percentage. So you think that the mitochondria takes up some calcium when calcium is released during the transient?
Yes.
Do you think they can shape the transient? Give me a percentage. They take up, I don't know, 10% of the transient, of the calcium released during the transient, 5%. What do you think?
Then we arrive to a very critical point. I mean, the estimation that amount of calcium taken up by the mitochondria in wild-type condition, estimation not mine, but done by, for example, the Baylor, by Rios, and so is that the mitochondria in skeletal muscle fibers of the mouse, of the rat, of course. I mean, this is the work just done on rodent muscle fibers. No? Is in the order of 5%, so it's rather little, no? In wild type. No? In our condition, we have at least 2 times taken up, no? So it becomes 10%. No? Mm-hmm. The mitochondrial volume increases at least 30%. So say from 20%, we arrive to 30%. Then the mitochondria goes more close to the calcium releasing unit, as you and Simone have shown. No? So then maybe even more. That, I mean, to which point we can arrive is difficult to say. Definitely, it's difficult to imagine that it's completely compensated in this way, because parvalbumin is very abundant. Parvalbumin is, the concentration of parvalbumin is in the rodent, mouse, and rat fibers, no? In the order of 1 millimolar. No? So it's, say, just to compare to troponin C, is 10 times more abundant than troponin C. Not to say about comparing to calmodulin. No? So to make a precise estimation, that is something very difficult. It's something that we want to do using the compartmental model, no? To be able to estimate precisely if all compensation can be due to mitochondria or something else can contribute. No? For example, removal from calcium to the extracellular, or calcium to the extracellular medium.
May I make my usual question? Of course, I am interested to know from experts, I am not an expert, how the different fiber types balance the different amount of mitochondria and and sarcoplasmic reticulum and triads, changing maybe how much? What is the difference in amount of mitochondria in for the same weight of contractile proteins in fast fibers and, let me say, type 1 soleus fibers?
Yes, that
There
is
are
a
the
very,
numbers
that's
somewhere.
it's a very good question, because the FDB muscle, the muscle which has become the reference for this type of study, is composed by 2X and 2B fibers. So all fast, all poor in mitochondria. Would be very nice to do the same in a soleus slow fiber, because mice are rather poor in slow fibers. Anyway, to be able to do the same type of measurement in a soleus slow fibers, where parvalbumin is very little expressed. Mitochondria are very abundant, and sarcoplasmic reticulum is not so much developed. So it's a rather different situation in a slow fiber.
I would like to have the opinion of Hood Levenburg on this. How muscle fibers that have a lot of mitochondria are able to maintain the calcium at the needed low level, and because mass fatigue is very soon in fast fibers, but not in slow fibers. I think that there is some basic missing interest or information, or there are problems in performing the analysis in collecting good numbers. But I continue to be curious on this.